We will give a presentation about affinity maturation. First, we will explain the definition of affinity maturation. The black shapes represent the antigen and the red shapes represent the antibody. When antibody binds to an antigen, various immune reactions are triggered, and the ease with which they bind is called affinity. It is known that antibody affinity gradually increases over time after exposure to a pathogen. This process, in which antigen-stimulated B cells undergo somatic hypermutation and selection to progressively increase their affinity for the antigen, is called affinity maturation.
Next, I will explain biological role of affinity maturation. There are two main roles. First, I explain enhancement of antibody binding. It ensures that the antibodies produced by B cells develop a higher affinity for the target antigen. This improvement allows the immune system to capture and neutralize pathogens effectively, even with lower concentrations of antibodies. Second, improvement of immune response. Affinity maturation helps the immune system respond stronger and faster when the same pathogen appears again or stays in the body. It also creates high affinity memory B cells, which are key for long-term protection.
Next, we explain which type cells are required. The main ones required are germinal center B cells, TFH cells, FDC cells. Germinal center B cells undergo somatic hypermutation and select high affinity clones. TFH cells promote B cell survival, differentiation, and high affinity selection. FDC cells retain material and present antigens to B cells. In addition, there are TBM cells, TFR cells, CDC cells that provide support and regulation.
Affinity maturation occurs in the germinal centers of secondary lymphoid organs, mainly the lymph nodes and the spleen. These structures appear after B cells are activated. The germinal center has two major zones. The dark zone is where B cells proliferate and accumulate mutations. It's called somatic hypermutation. The light zone is where high affinity B cells are selected with help from T follicular helper cells. B cells move back and forth between these zones. By repeating mutation and selection, they gradually improve their antibody affinity, and finally, B cells become plasma cells or memory B cells.
I will explain the mechanism of affinity maturation. Affinity maturation occurs in general centers of secondary lymphoid organs, which are divided into a dark zone and a light zone. In the dark zone, B cells undergo somatic hypermutation. They express an enzyme called AID, which converts cytidine to uracil in the variable region of immunoglobulin genes. During error-prone DNA repair, many point mutations are introduced, generating B cells with higher, lower, or unchanged affinity. Next, in the light zone, clonal selection takes place. Follicular dendritic cells present antigens, and B cells compete to capture them. Only high-affinity B cells survive, while low-affinity cells undergo apoptosis. These selected B cells can return to the dark zone for further mutation, repeating the cycle and dramatically increasing antibody affinity.